So let's talk about stomach motility for a little bit. The stomach is a bag, a reservoir for food that's important for processing it, digesting it, and in the regulation of the movement of food through the digestive tract. Now here's the anatomy of it. You have the fundus up the top, and then the body, which has this oxyntic glandular mucosa in it. It also has this pacemaker zone in the greater curvature of the stomach, and then below that, below the body, the corpus, below the corpus is the antrum with the pyloric glandular mucosa, then the pyloric sphincter, or the pylorus that separates the stomach from the duodenum. So the functions of stomach motility are here. Like I said, it's a reservoir for large volumes of food, and it's able to distend to accommodate quite a significant amount. There's a decent amount of motility, particularly towards the bottom of the stomach, where it grinds up the food and then regulates the emptying of the gastric content into the duodenum. The stomach, like the rest of the GI tract, has these slow wave phenomena that is the electrophysiology that underlines the motility of it. And these slow waves are initiated in the pacemaker zone, which I pointed out just before, in the gastric wall, in the greater curvature of the gastric body. And these will send pulses out, sort of about 3 every minute, that starts the contraction. This figure here shows both the electrical impulse and then the corresponding contractile force that goes along with these membrane potential changes. It's triphasic. You have a rapid initial depolarization followed by a plateau and then repolarization. It's similar in shape to like an action potential you would see in cardiac tissue, where obviously the temporal and magnitude resolution of this is different. Here we only have a depolarization of about 40 millivolts from minus 80, minus 90 to about plus 40, and they go for about 6 to 8 seconds, whereas in the heart the depolarization is in the range of 90 millivolts, and these are for milliseconds. So these are smaller and longer. And then you can see the contractile force that goes along with it. The longer the action potential, the slow wave propagates, the more contractile force you get. And in order to get any kind of contraction at all, the membrane potential has to break this threshold for contraction. The modulation of gastric slow waves is by acetylcholine and gastrin, which will increase the amplitude and duration of these slow waves, and then norepinephrine decreases the extent of depolarization and the duration of the plateau. In the antrum and pylorus, action potentials are superimposed on these slow waves, and these will increase the force of contraction, as we see previously in the overall view of motility. This figure separates the stomach into several different bits and looks at the electrical activity in each of the sections. So these different sections are the fundus, the orad corpus, the mid corpus, the caudate corpus, the orad antrum, and the terminal antrum. If you take cells from these and put an electrode in them, you get and record voltage changes, you get these traces that you see here on the left. And as we go through it, you'll see how different parts of the stomach then contract. If we go and start at the top with the fundus, the electrical activity there is pretty silent. And that's because the fundus's main role is as a reservoir. This will distend and expand as more and more food goes into the stomach, and it just basically allows a larger volume without any real contractile movement to it. Then as we go down to the variorid corpus and the orid corpus, you start to see these slow waves originating. You can't hardly see them in the orid corpus, in the variorid corpus, but the orid corpus, they're quite well defined. And then as you go down through it, you'll see that these slow waves become greater and greater, and then down into the antrum you start to see the action potentials showing on them, and then the pyloric ring has many on the top. And this is consistent with, as you go further and further down into the stomach, the contractions become greater and greater, and so the bottom of the stomach is where most of the grinding and mixing occurs. And the pyloric sphincter is obviously the point at which flow out of the stomach is regulated at. Now, you might notice that the slow waves have different frequencies as you go down, and this is just because what these recordings are isolated muscle cells from the specific strips. And similar to the heart, the pace is set by pacemaker cells, the interstitial cells of Cajal, and they are found in the body, in the corpus of the stomach, and they set the rate. And you can see that you see 3 of these slow waves in the corpus. And then as you go down, now you've taken these cells out of the stomach, and they set their own rate of firing. And similar to the heart, if you were to remove the sinus node, then the ventricular myocytes or the atrial myocytes will fire at their own rate, but slower. And the same kind of phenomenon is here. These cells do fire these slow waves off, but at a slower rate than the place that has the pacemaker cells. And so when it's all together, all these slow waves will be synchronized because of the pacemaker zone in the stomach and the intact structure of the heart, of the stomach. So gastric motility has 4 functions: filling, storage, mixing, and emptying. So the stomach is a big bag, and the empty stomach has about a volume of about 50 mils, but this can expand to greater than 1 liter. And this volume increase is not paralleled by a similar increase in intragastric tension, because the stomach has plasticity, whereas the stomach's smooth muscle cells can be stretched within limits without a change in tension. This is different again from cardiac tissue. As you increase the stretch, you increase the tension. Here, this is the same. They have a certain amount of plasticity to the smooth muscle cells. But there's also receptive relaxation going on in the stomach as well. Filling will cause a reflective relaxation of the fundus and the body of the stomach, so that the walls are able to stretch and prevent increase in tension. And this reflex is mediated by the vagus nerve and the neurotransmitters vasointeractive peptide and nitric oxide. And you can see here what happens if you cut the vagus nerve to the stomach. Under normal conditions, as you increase the volume of the stomach, the intraluminal pressure doesn't change, because the vagus nerve then is putting nitric oxide and VIP into the stomach and relaxing it, and so the pressure maintains. But you cut the vagus nerve, you start increasing the volume in the stomach, and the pressure increases because you don't have this relaxing reflex from the vagus. Gastric storage is mainly due to expansion of the fundus. The muscle layers in the fundus are thin, and there's very, very weak peristaltic contractions or motility, and so the fundus is really the part of the stomach that's involved in storage of unmixed food. Gastric mixing, however, happens towards the bottom of the stomach, and you have sort of three separate events that happen consecutively. You have propulsion, where a peristaltic wave starts at the top of the antrum and flows down towards the pylorus, and this will push the bolus towards the pylorus until it hits it, and then this peristaltic wave continues and grinds all the food stuff into a paste, and this pressure also will empty some of the gastric contents into the duodenum. And then you have retropulsion as this peristaltic wave continues towards the pylorus. The bolus that isn't pushed through the pyloric valve is then pushed back into the proximal stomach. Gastric emptying is stimulated or controlled by antral peristaltic contractions that drive the bolus towards the pylorus, and then the pylorus will relax briefly to allow a small amount of the gastric milieu to be pushed into the duodenum. And this is controlled by neural and humoral regulation, humoral and hormonal fine regulation from factors that are produced in the duodenum and the jejunum, and also it can be controlled by factors outside the GI system as well. So the bipyloric valve, just to reiterate, regulates the emptying of gastric content, and it also prevents regurgitation of duodenal content into the stomach. The relaxation of the pylorus is by VIP and nitric oxide, and the constriction is caused by excitatory cholinergic vagal fibers, and there are sympathetic fibers that also affect the pylorus, and hormones like cholecystokinin, gastrin, GIP, and secretin all have effects on the pylorus to regulate its activity and thus the emptying of the stomach contents. So gastric factors that affect the pylorus, the volume of chyme stimulates motility in the stomach, and the fluidity is another aspect that allows more rapid emptying. Obviously, fluids are able to be pushed through the pylorus at lower pressures, and so they easier flow through. Here are a summary of the duodenal and jejunal factors that regulate gastric emptying. So increase in acid in the duodenum, increase in fats in the duodenum, hypertonicity and amino acids. These will all decrease gastric emptying through intramural intrinsic reflexes through chemoreceptors, but also through the central nervous system. Sympathetic activity will reduce gastric emptying, and a decrease in parasympathetic will also decrease gastric emptying too. The increase in acid in the duodenum causes a secretion of secreting, and this decreases gastric emptying, and the increase in fats then release cholecystokinin and GIP, which are the mediators for decreasing gastric emptying by fats. pH in the duodenum and the jejunum is also something that reduces gastric emptying. So if we look at this electrophysiology graph of the activity of the gastric antrum compared to the mid-duodenum, you see under normal conditions the antrum is pulsing and then contracting, and then as you start to infuse acid into the mid-duodenum, the duodenum starts to become more motile. You see these electrical waves spikes of contractions are going along with this, and concurrently with this, the antrum then becomes much more electrically quiet, showing that the contractions are prevented. And this is because of the acid going into the duodenum results in the secretion of secretin, and then secretin will lower the motility of the gastric antrum and therefore reduce gastric emptying. Can you reiterate in these effects? Fatty acids in the duodenum and jejunum stimulate the secretion of cholecystokinin and gastric inhibitory protein, which inhibits gastric emptying. Hypertonicity, as chyme comes into the duodenum, it's usually hypertonic. Water is then dragged into the duodenum. These osmoreceptors sense that and then decrease gastric emptying via neural and humoral pathways. Peptides in the duodenum will stimulate gastrin from G cells in the antrum and duodenum, and this increases the constriction of the pyloric ring, and that will prevent gastric emptying. The peptides and amino acids can also promote the release of GIP and cholecystokinin, and this, as we've seen, inhibits antral contractions, and this will inhibit gastric emptying as well. And finally, dissension. When you have a food bolus or a chyme bolus in the duodenum, the GI tract senses that, and this will then intrinsically inhibit gastric emptying. Outside the digestive system, your emotions can alter gastric emptying, and or may even stimulate it. And intense pain is increased sympathetic activity, and this will result in the inhibition of motility. These are the consequences of controlling the emptying of the stomach, basically what the body is trying to do here. So if you control the emptying, fat is not emptied into the duodenum faster than it can be emulsified by bile components. So essentially you want to take in all the nutrients that you put in, and if you just flood it with a load of gastric digestive juices, there's too much for the duodenum to absorb, it's just going to go through and end up in the feces. And so the controlled regulation of that ensures maximal uptake of fats. The acid generated in the stomach is damaging to the epithelial layers of the duodenum, and so you want to be able to ensure that the acid that comes in is neutralized as efficiently as possible. And so by regulating the controlled emptying of the stomach, you can assure that there will be no damage to the epithelial layer. And other components of chyme don't enter the small intestine at a rate faster than can be processed by the small intestine. So essentially the take-home message here is the stomach can't just flood the small intestine with acidic contents. It needs to do it at controlled small amounts so that it can be neutralized and absorbed, so you can take all the nutrients necessary for homeostasis.